I see that it's 3 o'clock, so I will get things started for us. Welcome, everyone. Thank you for joining today's HPL seminar. Today, we'll be hearing from Dr. Kelsey Lucas. Dr. Lucas is a newly appointed assistant professor in biological sciences here at the University of Calgary. She completed her undergraduate degree in biology at Roger Williams University. She then did her master's and PhD in organismic and evolutionary biology from Harvard University. Her graduate work focused on the mechanics of force production by swimming fishes. Following, she was an NSF postdoctoral research fellow at the University of Michigan, where she explored the impacts of climate warming on fish physiology and the links between fish morphology and environmental characteristics. Overall, her research interests include the morphology and biomechanics of aquatic animals. She primarily works in fishes, but has also contributed to work in jellyfish. One of her proudest scientific accomplishments is being a mentor for students interested in research. Moving forward, she is excited to launch her lab here at the University of Calgary. As a fun fact, her hobbies include distance running, gaming, hiking, and photography. As a reminder, I will first turn it over to Kelsey for her presentation, and then we'll have the question and answer period to follow. And at that time, I'll remind everyone how to submit the question. So with that, Dr. Lucas, you're welcome to start your presentation when you're ready.
Great, thanks. Okay. Great, thank you for the introduction, Emily, and thank you and Doug for inviting me here today. Thank you all for being here. As Emily said, I'm a new faculty member in the Department of Biological Sciences here at the University of Calgary, where I work in fish biomechanics. So today's presentation is going to be a little bit of a change of pace for the HPL seminar series, but I really appreciate this opportunity to introduce myself to more of the Calgary biomechanics community. And I also welcome further discussion or opportunities to collaborate. So today I'm going to talk to you about some of my graduate work in fish biomechanics and how this leads into the projects I'm envisioning here at U of C. Before we get into that, I do want to zoom way out and provide you with a little bit of context. So while most of us think about life on Earth, we tend to think about the things around us here on land. But this perhaps is a little bit of a limited viewpoint because 90% of the world's habitable volume is actually in aquatic habitats. These habitats are host to a huge diversity of life. Just looking at the animal kingdom, about half of the invertebrate phyla are exclusively marine or saltwater. And looking at vertebrates, there are about 32,000 species of fishes. That's more than mammals, birds, reptiles, and amphibians combined. So all of this is to say that to understand life on Earth, we really need to understand what's happening in aquatic systems. It's also really important for us to recognize how different living underwater is from our experience here on land. Most obvious is the difference between oxygen content in air and water. So for equal volumes, air and water, the air will have 35 to 40 times more oxygen available than the water will. But air and water also differ in their physical properties. Water is 1,000 times as dense as air, 100 times as viscous or as thick. And so these physical properties can actually impede locomotion. As an example, drag in aquatic systems significantly limits aquatic animals' ability to accelerate. So together, this means that generating flows for swimming and feeding and tolerating flows from the environment become huge challenges for marine and aquatic organisms. With this in mind, my work is themed around these big questions. Fundamentally, I want to know how aquatic animals swim and feed from a very biomechanical perspective. But I'm also really interested in the context of these biomechanics, thinking about things like how the physics of movement is related to body plan evolution, and how biomechanics inform and constrains what aquatic animals are doing in their environment. And looking at the big picture, I'm also really curious about what the role of biomechanics is in aquatic ecology relative to other factors. So mainly these days I work with fishes, though I do have a line of collaborative work in jellies. Fishes are a great system for understanding form and function relationships in aquatic systems. As I noted earlier, there are about 32,000 species of fishes. And of course, that leads to a huge diversity in forms and ecological roles. But within that diversity, there's also a lot of repeated patterns of shapes and ecologies. And if you were to look at the saltwater fishes on the left side of the screen and compare them to the drawings of the freshwater fishes on the right side of the screen, you probably can pick out some repeated body shapes and body and fin shapes. Now, folks have been interested in fish swimming for a really long time for both biological and engineering purposes. To give you a sense of the scope of this, for example, in 1809, Sir George Cayley was trying to design a boat that was faster, and his design was based off the streamlined body shape of a trout. So very long history here. Over the last couple centuries, we've arrived at a pretty well-established framework of hypotheses that link fish body shape and swimming movement patterns to different swimming abilities. So if we, as an example of this, if we take a look at the fish in the top left part of the screen here, the tuna, it has a shape that's very similar to an American football. And that body shape combined with the crescent moon-shaped tail and movements localized to that tail are thought to allow the tuna to swim at very high speeds with very high energetic efficiency. In addition, swimming is involved in many critical fish behaviors, such as migrations or foraging and predator escapes. And so it's also thought that certain swimming abilities have been selected for and have influenced the evolution of fish body plans, as well as governing fish ecological roles. Continuing with our tuna example, its high speed and efficiency are very well suited to the lifestyle of a large ocean migrator. So although we have this very long history of fish biomechanics work, and this particular version of a framework of hypotheses linking body shape, movement, swimming, and evolution and ecology dates to Paul Webb in the 1980s, this framework has very rarely been interrogated, and that's mostly due to technological limitations, which I'll get to in a little bit. Before I move on, I do also want to note that these biomechanics aren't occurring in a vacuum. A lot of other processes are involved in swimming ability and fish evolution and ecology, things like physiology, as well as processes like geological history, species interactions, and invasions. So today, I want to focus on how I've been exploring fish form-function relationships. And in particular, I want to talk about some of my recent work revisiting some basics in fish swimming mechanics. As part of this, we'll look at some historical papers and common assumptions about fish swimming, and we'll look at some new methods I've developed for estimating swimming forces at high spatial and temporal resolution. And in addition, we'll think a little bit about what these methods have revealed about a particular mode of fish swimming. After that, we'll take a foray into future directions and stuff I'm thinking about in fish biomechanics and its ecological context, some directions I'm interested in pursuing here at U of C. Now, before we get too deep into the mechanics, I do want to quickly review some of the basics of fish swimming, just so that we're all on the same page. For this talk, I'm going to be focusing on one particular type of fish swimming called body caudal fin locomotion. Now, body caudal fin locomotion, as the name implies, involves the movement of the body and the caudal fin. And in this form of motion, fish swim by passing a sinusoidal wave of bending down their bodies that travels from the head to the tip of the tail. If we were to take snapshots of this swimming video, trace the midline of the fish throughout its swimming motions, and then stack all those midlines on top of each other, you can see that you would be able to see that the amplitude of this sinusoidal bending wave is increasing as it travels from head to tail. Now, I mentioned that because the characteristics of that waveform are used to categorize body caudal fin locomotion even further. And in particular, we tend to divide body caudal fin locomotion into two groups. These are the anguilliform locomotion, where most of the body is oscillating at a very large amplitude. And this is characteristic of fishes like eels and lamprey. And at the other extreme, we have carangiform swimming, where the posterior part of the body is oscillating at a large amplitude, but not the head and anterior body. This is very common of things like the bluegill sunfish that on the previous slide, as well as things like trout. And for today, I'm going to be focusing on this carangiform swimming mode. Now, before we move on, we do have one more term we need to know about, and that is tail beat cycle. So the tail beat cycle, you can think of as a unit of fish locomotion. It's roughly equivalent to a stride in terrestrial systems. Over a tail beat cycle, the tail beat cycle is defined by the movement of the tip of the caudal fin, and refers to when the caudal fin is at its most lateral extreme or peak excursion on one side of the body, that then moves to the other side of the body, to the other side's peak lateral excursion, and then back to its starting point. Now, for today, I'm going to be focusing on steady locomotion, where the fish is swimming in a straight line at a constant speed. And during steady swimming, net forces over the whole body and tail beat cycle sum to zero. But instantaneously or on specific regions of the body, forces may not be zero. So the net is zero, instantaneously may not be. Now, between the 1930s and 1970s, there were a lot of really important and famous foundational works in fish swimming biomechanics. For example, Lighthill's 1975 mathematical biofluid dynamics textbook. Now, a lot of these works focused on trying to link different movement patterns to force production. And kind of synthesizing a few of these and summarizing, these works in general propose this idea of separation of thrust and drag during steady carangiform swimming. Now, by separation of thrust or forward force and drag or force opposing locomotion, this is meant as both a spatial separation and a temporal separation. Spatially, we tend to think of the anterior or trunk region of the body as the drag region, whereas the tail fin, the caudal region, is the thrust producing component of the body. Temporally, drag and thrust also act differently. Drag is delivered continuously, so the trunk is acting as a continuous dead weight at the front of the body, whereas the caudal fin is producing thrust and pulses as it beats back and forth. So we have this idea of both spatial and temporal separation in thrust and drag. Now, this separation idea comes from thinking about positive pressure. For this to explain what I mean by that, I want to start by looking at the caudal fin, because I find that this tends to be a little bit more intuitive. So if this is a fish here swimming towards the top of the screen with its tail moving from the right side of the screen to the left, as the tail moves to the left, it's pushing on the fluid there, creating an area of positive or above ambient pressure in the space in front of that fin's movement. That positive pressure pushes outwards and pushes back on the fish's tail, acting as a pressure-based force. Pressure-based forces are always acting perpendicularly to a surface, so the orientation of the caudal fin next to that region of high pressure is important for directing forces. And in this case, the force has both a forward-facing or thrust component as well as a lateral component. So now that we kind of have a sense of what pressure-based forces are, we can think a little bit more about the fish's head. Now, the fish's head and interior body are thought to work a bit like an airfoil or a streamlined shape and flow. You can think of this shape here, this gray shape here, as being like the cross section of an airplane wing. What happens here is that as flow approaches the fish's body, And when it hits the head, it needs to divert to go move around the left and the right sides of the body. And when the flow diverts there, you end up with an area of positive pressure directly in front of the fish. That area of positive pressure, of course, pushes on the fish's body, opposes its forward locomotion. So we can call this a positive pressure pushing drag force. So with all of this in mind, you can kind of see where the idea of thrust and drag separation comes from, where you have this continuous positive pressure drag force acting on the head, and positive pressure thrust forces being produced as the tail beats back and forth. And while all of this sounds sensible, it's never quite sat well with me, because this model is actually leaving out negative or below ambient pressure. So to understand negative pressure, I want to again start by looking at the caudal region. Here we talked about how the fish's tail is moving from the right side of the screen to the left and pushing on the fluid on the left and creating that area of positive pressure. But at the same time, that tail fin is vacating space on the right side of the screen. And that leads to an area of negative or below ambient pressure alongside the trailing side of the fin. That negative pressure acts as a suction type force pulling on the fish's body. And on the caudal fin here, again, that the alignment of the fin in these pressure gradients affects which direction force is directed. So in terms of separation of thrust and drag, this doesn't really seem that big of a deal, because you still can imagine the negative pressure pulling thrust being produced as the tail moves back and forth. Where this becomes more problematic is when you start thinking about the anterior body. Now, if we're comparing the anterior body of a fish to an airfoil, the analogy that we've looked at so far is actually incomplete. So we talked about how flow arrives at the front of the airfoil and splits and diverts around the left and right side of the airfoil and creating an area of positive pressure. But the streamlined shape of an airfoil accelerates fluid alongside the body and leading to negative pressure along much of the length of the airfoil. This negative pressure, of course, pulls outwards on the fish's body, outwards on the fish or the airfoil's body. And so you end up with these suction-based forces acting along much of the length of the airfoil. Again, pressure-based forces act perpendicularly to a surface, so anterior to the point of maximum width, where a surface area is tilted slightly forward. These forces have a slight thrust component or forward-facing component. And posterior to the point of maximum width, these forces are pointed slightly backwards, leading to a slight drag component. Again, what this tells us is that the analogy of the fish interior body as an airfoil may be incomplete. And even more worryingly, this suggests that thrust and drag separation may not be as clean as we expect. So we've got some ideas to reconcile here, and maybe this is starting to sound a little far-fetched to you. After all, I'm raising a question about things we've often taken as fact for at least the last 50 years. But I want to note that this idea isn't entirely unprecedented. In fact, Dubois and colleagues, who are contemporaries of Lighthill, measured pressure along forces and measured pressure along the bodies of freely swimming fishes. To do this, they embedded catheters in the skin of a fish at all the locations indicated by these symbols here. These catheters were connected by hollow tubes to a bunch of pressure sensors. And then this whole fish and tubing setup was placed in this flow tunnel, which is essentially a swimming pool draining down the side of a cliff. So clearly, this is a set of experiments with a lot of limitations. They are highly invasive. In fact, Dubois himself is actually a lung surgeon and not a fish biologist. These experiments were also limited in their resolution because you only can make a measurement where you've surgically implanted a catheter. And the whole setup was prone to electronics failures. I think it's pretty telling that in one of their papers, Dubois and colleagues explicitly state that the measurement of pressures is not easy. Now, even with all of these limitations in mind, Dubois and colleagues were able to measure some mean pressures on the fish's body over the course of a tailbeat cycle. And what they found is a pressure profile that matches the airfoils pretty well, with positive pressure on the fish's head and negative pressure along much of the length of the body. They also predicted in this series of papers that this will lead to a combination of drag and thrust forces, with drag forces from positive pressure on the head, thrust forces from negative pressure along the interior part of the trunk, and drag forces from negative pressure on the posterior half of the trunk. So again, we have some stuff we need to reconcile here. We have this idea of thrust and drag separation, but we also have these negative pressures. And the question is, how do all of these fit together? Well, to answer that question, we need to know how and when forces are actually being produced on specific parts of the fish's body. But we run into another challenge here, and that's that we can't measure forces directly underwater. To give you a sense of why, I want you to think about how many of you measure forces in terrestrial systems. You'll use tools like a force plate, a solid object that you're pushing off of. We don't have those solid objects underwater, and so measuring forces becomes quite a bit more challenging. Now, there are some flow visualization methods that let us look at flow structures downstream of a swimming fish and make some estimates based on the momentum of those structures of how much force is being produced on the fish's body. But those methods only provide very coarse estimates of forces over large swaths of the body over large periods of time. And so they just don't have the resolution to be able to answer this question. And that's why it's not been really been studied since the 1970s. Now, we could estimate forces from pressure. Mathematically, force is equal to pressure times the surface area that pressure is acting over. But again, we saw that those Dubois-Stiles experiments are fairly impractical. So a lot of my graduate work was focused on overcoming this technological barrier, trying to develop methods for estimating forces at high resolution. Now, these methods have the workflow of these methods is pictured here. I start by measuring water velocity around a swimming fish. This is actually fairly straightforward to do, and we'll talk about what this looks like in a minute. After I have those force velocities, I can apply a set of equations from fluid dynamics called the Navier-Stokes equations. These describe how water moves, and among other things, they relate water velocity to pressure. Once I have those pressures, I can then multiply them by the surface area they're acting over and get an estimate of force. But before I show you what this looks like in practice, I do want to mention that I've completed a pretty extensive set of validations on this method using a physical modeling system where I was able to directly measure forces acting on the physical model using a force torque sensor, while simultaneously using flow imaging techniques and estimating forces using the workflow I just showed you. And I'm happy to answer some answer questions about the technical details of this later on. But the short version for right now is that for fish-like swimmers, this method does pretty well. So in practice, this is what the process looks like. I start by measuring water velocity using a flow visualization technique called particle image velocimetry. In this process, we swim a fish in a flow tunnel, which you can think of as an underwater treadmill. In this image of the flow tunnel, the water is moving from the left side of the screen towards the right. I then suspend a bunch of neutrally buoyant particles in the water and light those particles up in a thin slice through space using a couple laser light sheets. Now, once I convince the fish to swim in the light sheet, I can film from below using a high-speed camera and then track the particle and fish movements using a computer. So the results from this look something like this. On the left side of the screen here, you can see the raw PIV video. You can see the fish is brightly illuminated and that the tracer particles are showing us how the water is moving around the fish's body. Once I track the fish's motion and the particle motion, I can calculate a velocity vector field that shows us both the magnitude and direction of the flow around the fish's body. I do want to note for this vector field on the right side of the screen that I've subtracted out the net flow. So that way the rotational structures or vortices downstream of the fish's body are a little bit more visible. This is why you see arrows pointing upstream, even though the flow is moving from left to right. Okay, so once I have these velocity vector fields, I can calculate pressure fields through the Navier-Stokes equation. Navier-Stokes equations, I do this using an algorithm by my collaborator, John DeBerry. And the results of that look something like this, where you see a heat map of pressure in the water around the fish's body, with red colors being positive pressure and blue colors being negative pressure. Once I have that pressure field, I can multiply it by the surface area it's acting over. And that gives us a set of force vectors showing us both the magnitude and direction of the forces acting around the fish's body. So now that we have these high resolution methods, we can start revisiting the historical works and try to see if we can reconcile some of these hypothesized mechanics. Now, starting with the mean pressure over the tailbeat cycle, or the equivalent of what Dubois was measuring, we actually find pressure profiles that are very similar both to Dubois's predictions, as well as the pressure distribution on an airfoil. We see that there's positive pressure acting on the fish's head, negative pressure along much of the length of the body. Now, where things start to differ from Dubois is when we look at the instantaneous pressure data. So here on the left side of the screen, you can see the pressure on one side of the fish's body as it's swimming. On the right side of the screen, you see a chart of that pressure to give you a sense of the magnitudes we're talking about. The thick black line represents the mean pressure of the tailbeat cycle, and the thinner gray line represents the instantaneous pressure as the video is playing. Now you can see that there's quite a lot of variation in this pressure field over time that's not at all captured in the mean pressure trace. In particular, some of the features of interest we see here is that the position where the positive pressure on the head changes to negative pressure is moving anteriorly and posteriorly as the fish swims. We also see that in the caudal region that there's this point that there are points in time where instead of having the positive pressure predicted by the mean pressures, that the pressure along the fin is actually negative. Now for today, I'm only going to focus on the anterior half of the body, but I am happy to talk a little bit more about the caudal fins if that's of interest. So what does this mean for thrust production on the anterior body? Well, when we look at the forces, the first thing that jumps out at us is that much of the anterior body is actually producing thrust forces. What was really surprising to us, though, was just how forward on the anterior body some of this thrust is being produced. And there are moments in time where thrust is produced almost at the tip of the rostrum, as you can see from this snapshot. Now, that seems to be coming from seems to be the result of the fish's head movements. So if you think about carangiform swimming, I mentioned that there's a sinusoidal bending wave that's increasing in amplitude head to tail. Now, when we've been talking about airfoils, we've been talking about static, not moving airfoils. But a fish's anterior body pitches or rotates ever so slightly side to side. When we look at the mechanics on a pitching airfoil to try to get a sense of what's going on here, we actually do get some insight. So along a pitching, we're looking at a pitching airfoil, the flow that's approaching the airfoil is going to hit the airfoil at a different point rather than the front and center. Since that point where the flow divides to move left and right around the body is deflected to one side, the area of positive pressure also is deflected to one side. And that allows negative pressure to move a little bit more anteriorly on the other side of the airfoil. This leads to thrust forces acting very, very far anteriorly on the pitching airfoil. And this whole mechanical system in airfoils is called leading edge suction. If we take a look at the fish's pressure fields, we actually see very similar pressure profiles on the fish's head. You can see here that the pressure alignments are, the positive pressure here is deflected to one side, negative pressure has moved very far forward on the fish's body. Now, what's interesting about this to me is that if we're thinking about the separation of thrust and drag and the interior body being dead weight, you'd ideally want to have that dead weight not heaving side to side. Heaving side to side a little, pitching like this, just would incur extra additional costs. And this is actually described in quite a bit of detail in Lighthill's texts. But what this leading edge suction shows us is that the head oscillation may actually be beneficial to fishes, and so we probably shouldn't be discounting it. Okay, so now we know what the forces are like on the interior body. Let's think back to separation and negative pressure in more detail. We saw that negative pressure does matter, and it matters quite a bit. If you want a bottom line, the airfoil-like mechanics on the interior body produce about 28% of the body's total thrust. And if we think about negative pressure over the whole body, including the caudal fin, its negative pressure produces about 39% of total body thrust. So that's a pretty substantial deal, and it means that we can't be neglecting negative pressure. Having said that, I do also want to note that if we look at the total forces over the whole interior body, over the whole tailbeat cycle, we see that we see here that there are thrust forces being produced continuously, drag being produced in a little bit more of an oscillatory fashion, but still net drag producing all the time. And we find that the anterior body is net drag producing as predicted by the separation hypotheses. So what this means to us is that this idea of separation of thrust and drag is accurate, but only if we're looking at course timescales, the whole of the trunk, the whole of the caudal fin, and over the whole tailbeat cycle. Locally and instantaneously, this doesn't really hold up. And the swimming is, and so the bottom line here is that fish swimming is really complicated. However, I'd say that this is actually good news for us. We found that we don't need to rethink fish swimming biomechanics as a whole, although we did find some new interesting things about what's happening on a moment-to-moment basis. So we've spent a lot of time now thinking about the mechanics of swimming, and we found some interesting things out about fish. But where do we go from here? And more importantly, why do we care about these force distributions? Well, I want to remind you of this larger framework of hypotheses linking body shape, movement, and swimming to fish evolution and ecology. Today, we've mostly been hanging out in the upper right corner, thinking about swimming movements and swimming abilities. But we now have the tools to start interrogating the rest of this framework. And so my goal is to start putting these biomechanics that you saw today into a larger ecological context. In particular, I've been very interested in thinking about steady versus unsteady swimming. So when we steady versus unsteady swimming specialists. So when I talk about a steady specialist, these are fishes that are frequently swimming. Their swimming is generally directed in a straight line, and they're moving with a sustained velocity over a long period of time. Qualitatively, these fishes tend to be found in the water column of rivers or in open water. Local examples of fishes like this include trout and shiner. In contrast, some fishes are unsteady specialists. These focus on infrequent bursts of motion, either rapid accelerations in a straight line or turning behaviors. And qualitatively, these types of fishes tend to be found on the bottom or in highly structured environments. A local example is pike, and another example is the bluegill that we've been seeing throughout the talk. Now, I do want to note a major caveat here. These are very loose categories of swimming specializations. They're not super discrete. Think of it more as ends of a continuum. And second, these ideas come from temperate North American freshwater fishes, such as those found in the US and Canada. And these categories get a lot fuzzier once you start moving out of that system, particularly if you're looking at a coral reef. So just to reiterate and to help you visualize steady versus unsteady swimming, here are some examples of these behaviors. On the left side of the screen, you can see steady swimming, like body caudal fin swimming we've looked at throughout the talk. But this also includes swimming with other fins, like the pectoral fins in the bottom left here. And unsteady behaviors include things like accelerating or turning. So what's interesting about these steady versus unsteady specialists is that this also seems to be correspond to different morphologies. And these more hypothetical differences in swimming morphologies also seem to be connected to fish body plan evolution. There is quite a bit of evidence now that fish body plans are evolving in patterns that align with these two types of morphologies that you see here. Now, before I can tell you exactly where I'm going with this, you do need one more piece of background info, and that's about swimming in turbulence. Now, turbulence is 3D flow in natural environments. This comes from combinations of rotational flow structures like waves. And the key thing here is that turbulence in natural environments can be large enough to disrupt swimming force production on a fish's body and could cause the fish to spill and be swept away, risking injury or death, depending on the scale of the flow, if these turbulence disruptions are not corrected. Interestingly, fishes from high flow environments like rivers can stay upright and stay swimming at higher turbulence levels than fishes from low flow environments like a lake. So putting all of that together, qualitatively, fishes from high flow environments tend to be steady specialists, have streamlined bodies and small fins, and tolerate turbulence. Whereas fishes from low flow environments tend to be unsteady specialists, have bulkier bodies and large fins, and don't tolerate turbulence. This leads to this question: does something about a steady specialist morphology and movement patterns facilitate turbulence tolerance, allowing them to select high flow habitats? And this question is the big theme in a number of my current projects. So answering this question, I'm taking a number of different approaches. Of course, I need to start with the biomechanics basics. How do different swimming behaviors work, and do shape differences correspond to differences in force production? For example, we might think about things like the role of differences in body depth between the steady swimmer and unsteady swimmer, and how that might relate to streamlining and drag reduction. But I'm also now, with these high resolution force techniques, able to think about things like fin surface area and whether and how that relates to torque production. In addition, my collaborators and I have been beginning to think about more complex swimming behaviors. For example, recently we've been thinking about turning maneuvers by zebrafish. Now, in addition to these basic biomechanical approaches, I'm hoping to use the collaborations and data that I began in my postdoc work to start putting these mechanical studies into a larger context. And in particular, I'm really interested in determining how well these associations between morphology and habitat actually hold up. To do this kind of work, I rely on museum specimens, focusing on measuring morphological traits hypothesized to have specific swimming functions. And I also rely on a set of fish abundance and habitat surveys from Michigan's Department of Natural Resources. Now, with these two types of data sets, I'm hoping to be able to use some ecological modeling techniques like fourth corner analysis. This type of analysis allows us to associate species distributions with different habitat features from the survey data, and simultaneously try to provide some functional explanations of these species distributions using the morphological data set. Now, the benefit of this is that it allows us to move from thinking about shape and habitat qualitatively, as we've been throughout the talk, to thinking about it more quantitatively. And simultaneously, we can begin to explore the relative importance of swimming biomechanics versus other drivers of fish species distributions like temperature. Now, My postdoc was mostly focused on the state of Michigan, but I am hoping to finish up with this data set and expand to other regions, particularly across Canada. For example, here in Alberta, thinking about river systems from mountain headwaters into the plains is another really good system for testing these types of questions. So I think I'm going to leave it there for today. Are the key conclusions that I want to remind you about is that force production during carangiform swimming turns out to be quite a bit more complicated than we might have expected, and that we really need to be thinking about negative pressure. We also talked a little bit about how some future directions I'm interested in pursuing, placing fish swimming in an ecological context. With that, I'd like to thank a number of collaborators and colleagues. Thank you to funding sources, especially the U.S. National Science Foundation, which funded my postdoc. Thank you all again for being here today, and I'm happy to stick around for some questions.
Great, thank you, Dr. Lucas, for that super interesting presentation. I learned a lot. The floor is now open for questions. As a reminder, you can submit a question using the Zoom raise hand function. And if you are unable, please type your question in the chat box. And I see Amin, you have a question, so please go ahead.
Sure. Thank you, Kelsey, for the presentation. I'm going to understand the method that you use to develop the equation to calculate the pressure profile. You use Navier-Stokes equations, and there are some assumptions for that, the flow type. Is it turbulent or laminar flow that you had, or what about the viscosity of the water? Did you assume a viscous flow or unviscous flow? What was the assumptions that you make?
Yeah, so I think the short answer to your question, it would be the Reynolds number. The Reynolds number's in several thousands, like 10K. So it's
what was the number again?
About 10,000.
10,000?
Yeah.
Okay, so I cannot remember off the top of my head if that puts the flow type in turbulent or laminar. Can you tell me what was?
So when you work in a straight float tunnel, your goal is to have as laminar flow as possible. And so you can assume a certain degree of the flow being laminar. Certainly there is going to be some turbulence there.
Mm-hmm. Okay, thank you.
Art, go ahead.
My question is about basically my takeaway from your findings are that basically drag and thrust are dead in the sense that they don't help understand a lot of what happens in fish swimming. And I'm just wondering whether there is life still to drag and thrust with appropriate new information. I guess where I'm coming from is for designing aircraft, I think people can mostly live with pretty simple equations. And then they have these things like airfoil shapes and propeller shapes and things like that. And those properties are pretty well known. And then you only need to do Navier-Stokes if you're, designing a whole new propeller shape or something. But largely you can understand and get away with just using some, I don't know, codified information about airfoils and then some relatively simple equations. So I'm wondering whether there's any hope for fish as well, because I think most people are not going to turn to Navier-Stokes at any time they want to solve or answer a question about fish locomotion.
Yeah, so the good news is there is hope. The separation of thrust and drag for carangiform swimmers, it does hold if you're thinking about long time scales. So it is a decent shorthand in some conditions. In addition, the way that we're applying the Navier-Stokes equations here is through an algorithm that runs off of PIV data. PIV data is extremely common in fish swimming biomechanics. And so, this algorithm was actually designed for biologists' use. And so one of the unique things about it is that it tallies pressure data using a median polling algorithm after calculating pressure along several paths through a velocity field. What that ends up meaning for us in practice is that you sacrifice a little bit of accuracy for this algorithm running extremely quickly. So for the few seconds long videos that I have here, you can go from pressure to force in about an hour once you tell the algorithm where the fish is in space.
Mm-hmm. And then I guess related to this, I guess people have been studying different gaits of fishes for a long time. And I think usually they talk about it in terms of thrust and drag. And I'm wondering, from what you found, do you have anything different to say about gaits? Like, was there anything that people misinterpreted in the thrust drag story that you can shed light on from your approach?
Yeah, so gaits is a very loose term in fish biology. So we will sometimes talk about anguilliform and carangiform swimming as separate gaits. They're really more different, more extremes on a continuum. So you can think of basically how much of the body is using undulatory versus airfoil-like mechanics. That's more of the difference between those. I think in my thought on this, and what several other folks have been working on in recent years, is trying to find new ways to define gait versus for fishes that captures the variation in fish locomotion more appropriately, and in ways that allows us to actually make meaningful distinctions, because things like anguilliform and carangiform are great shorthand for saying eel-like versus trout-like, but they aren't super useful for saying things about the mechanics. Mm-hmm. Or rather, they aren't as useful as saying things about the mechanics. There are differences, but they aren't as useful as you might expect.
Great, thanks.
Doug, please go ahead.
Neat stuff, Kelsey. I had a different question, but Art's question about gaits sort of really struck me as an intriguing thing. So tuniform swimmers that sort of mostly just lock their nose still and beat their tail in a simplistic sense anyways. Do we know anything about them in terms of suction forces and what they're doing with their noses versus their tails in terms of generating thrust?
So nobody's tried it yet. One of the challenges is it's with tuna, especially. So tuniform, for those of you who aren't super familiar with fish vocabulary, tuniform swimming is a specific type of carangiform locomotion that tunas use, where the movements are extremely localized to a very small caudal fin. Now, one of the issues of studying tuna is that they don't do well in captivity. And so measuring forces on them in this way is quite challenging. And so I'm not sure that anybody has gotten to that yet. I do know that George Lauder's lab was working with tuna in a massive enclosure in Rhode Island, but I haven't kept track of where that project has ended up. I would say my offhand guess would be that it would reflect something more similar to carangiform locomotion. I can tell you that tuna's heads, although we talk about all of the locomotion happening at the caudal fin, tuna's anterior bodies are heaving back and forth a little bit. So I could imagine some airfoil mechanics happening that way. That said, tunas have very large bodies. We think about tuna as like the cans that you buy in the grocery store, but tunas are like 6 feet in length. So the amount of recoil there is pretty small, just because they have so much mass. So they probably are using some airfoil mechanics. I would hesitate to say that that's the most important thing happening on them.
Okay, can I have a quick other question? So you were measuring forces relative to the ambient, positive and negative relative to ambient pressure. Is the pressure inside the fish relevant, the differential across the body wall, where you could see if suction forces are being generated on one side, that will actually affect the pressures inside the fish as well? Or is it just translating across and net zero in terms of forces across the fish, even though they might be oscillating positive and negative as well?
It's a really interesting question, and I can say that I sincerely doubt anybody's done that yet. It'd be really interesting to figure out and see, especially since that could affect properties of the muscle tissue and that kind of thing.
Yeah, I mean, you would think in terms of how the fish body moves, it's the difference between the environment outside it and the body of the fish. And so it's the gradient across the body, not just necessarily whether ambient pressure is positive or negative relative to what it is 50 meters away from the fish. Anyways, yeah, I'm sure.
Yeah, so it is a super interesting question. And one of the reasons why we were interested in developing this technique was because we were interested in trying to think about body stiffness variation over the fish's body, stiffness variation over the fish's body as it swims, trying to figure out what the forces are acting externally to the fish, how that relates to the amount of bending that's occurring and that kind of thing. There's still quite a long way to go with that, but it is definitely something of interest and something that I have some collaborators who are working on.
Cool. Yeah, actually, I had a question about that, but I'll be quiet for a while and let other people ask questions.
Michael, go ahead.
Sure. Thanks, Kelsey. That was really interesting. I had a question about the energetic cost of these different movements, and is it relatively constant if we were to go from like the anguilliform to the carangiform, or is there very broad kind of differences in the energetic cost if you like normalize to the mass of a fish or something?
Yeah, so we generally tend to call anguilliform swimming highly efficient. As an example, there are eels that using that kind of locomotion that perform migrations between the center of the Atlantic Ocean and coastal regions in North America and Europe. And they'll make these migrations over months without doing any feeding. So that swimming has to be super efficient in order for them to work. The downside of that efficiency is that this type of swimming is relatively slow. If you're thinking about a high performance system looking to maximize speed relative to mass, you're going to be using something more like a carangiform locomotion.
Great, thank you.
Walter, go ahead.
Thanks a lot, Kelsey. Very interesting. I'm always interested in mussels. So when I saw your fish swimming there, and a very little excursion in the front part of the body and big excursion at the back, I was wondering, is that big excursion in the back done by mussels, or is it more like a whipping action that you have very small movements to the front part of the body, and then the tail is just reacting elastically, and you really don't need any muscles in the back of the fin there? Can you explain to me a little bit what the mussels might be doing?
Yeah, so I will say it's going to depend a little bit on which fish you're talking about. So looking at something that's super specialized like the tuna, pretty much all of their muscle architecture is in the trunk portion of the body. And then they have these really intense tendons that run down the caudal peduncle and actually actuate the fin. In other fishes, there's much more continuous muscle over the length of the body. And beyond that, I actually think Doug would be a better person to answer than I would.
Okay, I'll talk with Doug independently. The other question that I had, and that might be a little bit weird, but we are in kinesiology and we try to help people and athletes swim really fast. So what would you tell a human swimmer to look for to go fastest? Or do you know, and I think now I understand a little bit why people can swim faster in one way to another. Would you have any idea on how people actually can locomote in the water the fastest?
I wouldn't have any broad recommendations other than saying that the reason why those shark skin swimsuits are banned doesn't have anything to do with the shark skin texture and has everything to do with how tight they fit. So work on your streamlining, I guess. The other thing I'd say is that if you were to race, if you were to scale everything by size, the body length, a fish is going to outperform a human every time.
I'm sure they are. We're not really made for swimming, but what always fascinated me is, you think we would use our fins, like our arms and legs to swim fast, but people, at least really good swimmers, they seem to be fastest by doing exactly what fish are doing. They just undulate.
Yeah, there's definitely, I don't know tons about how human swimming works, but they're definitely using different postures to try and take advantage of different flow characteristics for sure.
Yeah, and that actually was forbidden because there were swimmers, especially in the backstroke, that would stay underwater for entire 50 meters, take one breath, and then swim back again, 50 meters all underwater, just undulating. And now the rules are that you're not allowed to swim underwater with the undulating motion more than 15 meters. But that seems to be actually the fastest way for people to swim, except every once in a while you need air. And then the last question that I had is, what about that funny shaped crescent moon-shaped fin that your fish seem to have? Is there any advantage to having that type of a shape of a fin, or is that random?
Yeah, so that tuna tail, that's like a super crescent moon shape. The tunas are extraordinarily specialized fishes. And so I wouldn't necessarily generalize a lot of tuna swimming to most fishes. But that crescent moon-shaped tail is a bit stiffer than most caudal fins of fishes. And the idea is that shape basically has really good fluid dynamics for force production in a really efficient way without incurring a lot of costs as it's moving back and forth. Like, that's a very simplified answer, but
yeah, good. Thank you.
Doug, please go ahead.
Yeah, so I'm sort of going to ask a question as Walter did about this notion. When you watch swimmers, they dive in and they porpoise. And I was going to say, if they could hold their breath for longer, they could easily win the race because they really book when they're underwater porpoising. Just that notion of that type of movement, do you think they are taking advantage of negative pressures and pulling themselves through the body? Is that possible? Or do you think there are much more sophisticated nuances and sensory systems and motor control required to take advantage of pulling yourself by your nose through the water or underwater?
So I would say that negative pressure is involved, but I don't know if I'd go as far as to say that they're like deliberately tuning to make that like the most of what's going on. Yeah.
Okay. Yeah. So it's so just undulations like that don't inherently give you those.
So I'd say that those undulations work a lot. You do with those kinds of undulations, you are going to end up with areas of negative pressure, and those areas of negative pressure would be able to contribute to swimming force, either thrust, drag, or lateral, depending on where they're located. But I don't think I would necessarily say that they work like something like a lamprey, which uses huge swaths of negative pressure such that most of its swimming force is about sucking itself forward. I wouldn't say that humans are doing anything that extreme.
Right.
So
okay.
It has a role. I just don't think you'd necessarily call that like the biggest role.
All right. Thanks.
Yeah, just maybe following up on that, people can put fins on their feet and swim incredibly much faster. Do you think negative pressure will play a role there when you put some fins on your feet?
On a flipper, for sure, yeah.
Yeah.
Yep.
That would happen then. So it would be similar to what you see in the fish then.
Yeah. So the thing that came to mind for me with that is computational simulations of things like a whale tail. And with those, you do also see negative pressure on portions along those.
Thank you.
I'll jump in with a question. I feel like I see a lot of kind of those extraordinary fish videos where a school of fish swims in coordination. could you talk about that? And does that at all relate to the forces that they're exerting and experiencing, or is it coincidence because they're all moving towards a certain target? Or
oh yeah, that's a fantastic question. So the way that fish are able to do that is they're using a sensory system called the lateral line. And the lateral line is actually detecting changes in pressure around the fish's body and using that to kind of sense how flow is moving around themselves. So for sure, some changing your pressure gradients as you're swimming is going to be interfacing with that sensory system in some way. Now, there's been a little bit of work showing that that sensation on the head, for example, is kind of I guess the best way to put it is that it is paying attention to the pressure differences as they're swimming in the head region, and that it might be really important for the fish's sensory capabilities. But I think this idea of understanding how pressure is being developed and moving along the fish's body and how that relates to sensation and how fishes coordinate movement, I think is an emerging area in fish biomechanics. So I'm excited to see people pursuing that.
So just kind of following up on that, sorry for jumping in here. So, you know, there's fish obviously that migrate long distances. And when they do that, do they also swim kind of in a certain formation like the Canada geese, you know, that have this formation that supposedly is energy efficient for the geese in the back? And do fish do that as well when they migrate over long distances, that they swim in certain configurations that might save them energy?
Yeah, so this idea of schooling is something that folks are definitely looking into. One of the challenges is that unlike following a Canadian goose where you can just kind of like drive along and watch what they're doing, it's a lot harder to track what fishes are doing in the open ocean. So I think trying to figure out what's happening in a long ocean migration, specifically what the formations look like over that migration, I think that's pretty much an open question right now. In terms of, are some schooling configurations advantageous? I'd say loosely, yes. There's been some lab-based study and a lot of computational-based study trying to study different configurations of fish swimming in schools and trying to see if they can get different performance benefits out of that. And it does seem like that there is some.
Amin, please go ahead.
Yeah, so I noticed that the analysis that you performed was in 2D, in from top view, and you'll see all the motions and morphology change in the body in that plane. But what about other possible motion? For example, is there any spinning when the fish moves forward? Did you investigate any other direction?
Yeah, so it depends. Again, it's going to depend a lot on the fish and the type of motion. Anguilliforms have quite a bit of that type of rotation. So we call that type of rotation wobble. And so you see a lot of wobble in the caudal, probably the posterior half really of a lot of anguilliform swimmers. There's a line of work by Cassandra Donatelli and Eric Teitel looking at that, and that wobble does seem to impact different force production, like the amount of force production and swimming capabilities and that kind of thing. 2D, we're often in fish biology limited to 2D PIV because rather than 3D systems for a few reasons. One is that it's easier to do. Another is that 3D systems tend to have a much lower resolution than a traditional 2D PIV system, and finding some of these finer details can be a bit tricky in the 3D system. The last reason why we tend to avoid 3D systems is because it's very difficult to tell those systems where the fish is in space. It's a very non-trivial problem. And so 2D PIV tends to be the workhorse for that reason. The way we try to get around that is First, by either focusing on a swimmer that doesn't do a lot of things like wobble, or you can also just move your PIV laser, put it in different planes and try to do some kind of tallying up of different fields in that way. So we normally put the PIV laser at the mid-frontal plane, but there's no reason why you can't image with your PIV laser set up to be in a transverse section, for example.
Okay. Yeah, you know, one thing that concerns me is that, let's say you have like out of plane velocity that you don't measure, but when that like velocity hits inclined surface, it's going to create drag or thrust force in the direction that you study, and you have to ignore that, or yeah, you don't measure it.
Yeah, Emily, am I still capable of sharing screen?
Yes.
Okay, so yeah, let me just bring this up. So that's actually one of the major focuses that we looked on in our validations. So we were, what we did is positioned our PIV laser in multiple places around our model. Here I just have a sample set of data from a rectangular airfoil, but we also did this on more fish-like models, including ones that had a caudal peduncle built in, so that way the tip vortices on the model would be specifically be directed into and out of the flow for this. And what we found generally is that for fish-like swimmers at high enough Reynolds number, that we're still able to reproduce the shape, timing, and magnitude of force versus time trends in measured on the model system, even with some degree of out of plane flow. So in this specific model, the amount of flow that's happening right on the edge, this PIV laser is positioned 5 millimeters from the edge, right in the middle of the tip vortex. About 33% of the flow, if you look at the whole total flow magnitude, about a third of the magnitude is directed out of the PIV plane. What you're seeing here is that we are underestimating forces in that position, but we're still doing reasonably well. I wouldn't necessarily want to stake my life on a results from taken in this way, but if we're trying to get a sense of what forces are like in that area for the first time, this is giving us a pretty decent approximation that we've never been able to have before.
Thank you.
Are there any other questions from the audience?
Maybe just a very quick question. Has ever anybody tried to measure the forces on a fish directly, like you have them in a tank and you, I don't know, have them somehow glued to a three-dimensional force transducer on the back there somewhere in the midline, and then they try to swim, and then you measure the forces directly somehow like this, or anything like that? Has anybody ever tried that?
So I'm not aware of anybody who's done that for forces. The Dubois studies, one of the things I didn't discuss today is that they glued pressure transducers to the fish's caudal fin and tried to measure pressure that way. But I'm not aware of anybody who's done that with forces.
I know people have done that with fins and so forth, that you put a fin in a swim flume with a pin attached to it and force transducers. But yeah, as Kelsey said, I don't know if anybody's tried it with an actual fish.
I was just reminded of studies, like these insects, where they pin the insects on the little three-dimensional transducer, and then the insect tries to fly away, and you can directly measure the forces that they exert while they try to fly.
So having been in Michigan, where in the department that Paul Webb is emeritus of, I've heard some wacky stories of him trying to tether fish to do things like that. And his comment was that it didn't work very well. I will say that the last time he tried doing that was probably in the 80s or the 90s. Technology's increased quite a bit since then. I don't know if anybody's ever tried to do it since.
Yeah. Technology evolved, but the fish stayed the same, so they might just not really be amenable to that. Yeah.
Any other final questions? Okay, with that, please join me in again thanking Dr. Lucas for her presentation today. And thank you to the audience members for all the great questions and discussion. And I hope to see everyone again next week for next week's HPL seminar.